Today's Grand Rounds is part of the Institute for Clinical and Translational Research at Medical Grand Rounds and is titled COVID-19 Vaccines Past, Present, and Future, and will be presented by Dr. Kathleen M. Neuzil, Myron M. Levine Professor in Vaccinology, Professor of Medicine and Pediatrics, and the Director of the Center for Vaccine Development Global Health at the University of Maryland School of Medicine. Dr. Neuzil is an internationally recognized research scientist and advocate in the field of vaccinology. Throughout her career, Dr. Neuzil has conducted clinical and epidemiologic studies on vaccine-preventable diseases, yielding high-profile publications that inform policy decisions and public health actions. Dr. Neuzil's work has spanned dozens of low-resource countries with multiple vaccines, including influenza, rotavirus, human papillomavirus, Japanese encephalitis, typhoid conjugate vaccines, and most recently, COVID-19 vaccines. Dr. Neuzil is central to the domestic and global response to COVID. As a co-PI of the NIH-funded leadership group for the Vaccine and Treatment Evaluation Unit Network, Dr. Neuzil is part of the strategic team evaluating COVID vaccines and therapeutics in the U.S. and was part of the study team who designed the first COVID-19 clinical vaccine trial in the U.S. The CVD is likewise participating in COVID treatment and post-exposure prophylaxis trials. Dr. Neuzil also directs TIVAC, the Typhoid Vaccine Acceleration Consortium, with the goal to accelerate the introduction of typhoid conjugate vaccines into low-resource countries. She has more than 230 scientific publications on vaccines and infectious diseases. Dr. Neuzil's research capabilities are complemented by 20 years of involvement in domestic and international policy, including past membership on the U.S. Centers for Disease Control and Prevention's Advisory Committee on Immunization Practices. She is a member of the World Health Organization Strategic Advisory Group of Experts on Immunization. Please join me in welcoming Dr. Neuzil.
Unmute. Thank you very much for that. Am I still muted? No, I'm unmuted. Thank you very much for that introduction. You'd think I've never used Zoom before, which obviously isn't true. I am here today to talk about COVID-19 vaccines. I will give you the current status, although I have no doubt in the hour that I'm sitting here, if anybody's following Twitter or the New York Times, you will be more up to date than I am by the end of this talk. So I thought I'd just start with a cartoon that pretty much describes the way I feel right now. We were all looking to the end of 2020, and we've sort of been launched right into 2021 with this virus continuing to surprise us. I'll talk about some of the variants that we're seeing and some of the challenges that we have here heading into 2021. In all seriousness, though, this pandemic has really been a disaster. You know, it has caused unprecedented morbidity and mortality. It has shut down schools. It has shut down our economy. And here is a look as of this morning from the World Health Organization, where we have more than 111 million cases, more than 2.5 million deaths. And something that we're not used to seeing usually when we look at the World Health Organization global statistics, and that is the Americas here in yellow and specifically the United States leading the way in terms of morbidity and mortality. You know, you've heard a lot lately about the decline, and we are certainly seeing declines in the US, we're seeing declines in Europe, we're seeing declines elsewhere. But if you look at where we are right now and just move across the map, you know, we're still no better than we were in October. So yes, things are getting better. There is still a lot of disease out there. There is still a lot of transmission out there. Now, the other epidemiologic aspect I find fascinating is we're really not seeing other respiratory viruses. And I'll just make a couple comments here. But if you look at the way we track flu in the United States, and this shows select seasons over the last 10 years, and predictably, we see flu peaking sometime in December to March. And here we are right now with essentially no influenza activity. This is not only true here in the United States, it's true around the world. It was true this summer, our summer in the Southern Hemisphere. So a very intriguing epidemiologic outcome of the COVID pandemic, which may partly be due to control measures, but even in places where these measures haven't been fully implemented, and I would include the United States in that group, we aren't seeing influenza. Another way we track influenza is by looking at excess pneumonia and influenza cases. And I think this slide says it all. You know, if you look at our regular seasonal baseline with more deaths due to pneumonia and influenza in the winter than the summer, we call this the epidemic threshold, and this excess threshold are the pneumonia and influenza deaths attributable to pneumonia. And you just see here how COVID is off the scale. So we now call this pneumonia, influenza, and COVID mortality that we track. I will tell you, it's not 1918. If it were 1918, it would be completely off the scale and off my slide. But nonetheless, the 500,000 cases in the United States are a number of deaths we haven't seen since 1918 due to pneumonia and influenza. Now, fortunately, we're not in 1918, we're in 2021. And so therefore, we had the science, the technology, and the ability to accelerate vaccine development. We know it's a lengthy, risky, and expensive process. It was sometime last January where the genetic sequence was available to scientists at the NIH and elsewhere. And I first got involved in efforts to map out what the vaccine development program would look like, and specifically how we would test these vaccines in clinical trials. And when people ask me, how did we do this so fast? You know, it's been decades of investment in science and technology that allowed us to do this. It's because scientists understood SARS-CoV-1, understood MERS, understood seasonal coronaviruses, that we had laboratories with a head start on assays, that we understood the S-glycoprotein. Because mRNA vaccines had been developed for almost 3 decades, we were able to advance them quickly during this pandemic. We also saw unprecedented resources, your tax dollars, my tax dollars, that went into this effort and allowed us to assume success, have parallel development every step of the way. And finally, I witnessed leadership by the US government, leadership by the NIH in terms of partnership, bringing the regulators to the table, the manufacturers to the table, academics to the table, people with different expertise to work together towards a common goal, share lessons learned, share challenges, and really the regulators being involved from the beginning helped facilitate the speed of this effort. And in fact, science called the development of these vaccines the 2020 breakthrough of the year. So I believe last June, May or June, when I gave grand rounds, all I could really talk about was the hypothetical. And I actually spent a lot of time on the SARS-CoV-2 spike protein. I won't spend a lot of time on it today because I'd like to get to the actual vaccine data and a lot of the critical questions that are out there. But just to say that all the vaccines that I'll be talking about today, all the vaccines in late stage development in the US are in fact based on this spike protein and the idea that antibody to this spike protein will prevent the binding of the virus through this ACE2 membrane and prevent infection. So here we are. I also get my latest news from the New York Times. So as of yesterday, here is what we had globally: 37 vaccines in phase 1, 6 vaccines in early or limited use, and this includes the vaccines in the United States under emergency use authorization. So the Moderna and Pfizer vaccines are included in that category. There are fully approved vaccines in other countries, in China and in India. Now, just a reminder of where we were last June. I talked about how important it was that the regulators, the FDA specifically, have been involved in this effort from the beginning, put out guidelines in terms of development and licensure of vaccines to prevent COVID-19. And in fact, 2 days ago, updated these guidelines to include information on the variant strains and how new strains of COVID-19 vaccines would be handled. So just as a reminder, because I feel that we've been a little bit spoiled with some of the early efficacy results with the mRNA vaccines, is that the FDA specified that we should have a minimum efficacy of 50% with a lower bound of the confidence interval of 30%. So this is more in the range of what we see for influenza vaccines, for example. Because it is a public health emergency, we made the decision that all of the large phase 3 trials done in the US with sponsorship by the US government would have a minimal sample size of at least 30,000. We were told that we should use the CDC definition of COVID disease as a guideline for that primary outcome, with more serious outcomes as secondary. That there should be close follow-up of COVID positives, particularly early on. There was this concern about vaccine-enhanced illness. So when people were determined to have a positive COVID test, we should follow them carefully. And then there was a lot of emphasis put on enrollment of those at high risk for disease. So older age groups, people with chronic conditions, minority populations, so that the population in the trials would reflect the populations who are getting serious and fatal COVID-19. Now, we also looked at, what are the appropriate clinical endpoints and how might these clinical endpoints affect how we evaluate efficacy and the efficacy estimates we end up getting as part of these clinical trials. I won't go too much into this for the sake of time, but I would encourage you to read this paper, because we are seeing the consequences of people not understanding these concepts as they misinterpret data that are coming from these clinical trials. So let me explain that. When we talk about SARS-CoV-2 infection, we're talking about a spectrum of clinical illness that would go all the way from asymptomatic infection to symptomatic infection, which would include severe COVID and non-severe COVID. So depending on how you define symptomatic infection, how you define severe COVID, your efficacy estimates may change. We also had very close follow-up in these trials with collection of data on disease severity, obtaining samples, both blood samples and nasopharyngeal samples on anyone with a COVID diagnosis for, in some studies, 2 weeks, in some studies 3 weeks or more, to again fully characterize that COVID illness. So again, what you're seeing when you see data from these clinical trials is you've mainly seen the primary outcomes and some of the secondary outcomes. There will be a lot more that we will learn about attenuation of symptoms, about whether the period of viral shedding is positively impacted by these vaccines as we get more data from these trials. Now, the one aspect that we really haven't considered as part of these trials, although we would certainly have the ability to do so, is sequela in people with COVID-19, that not everybody has an acute self-limited infection, but we are recognizing more and more that particular symptoms of COVID may be persistent. And this is an article that appeared in JAMA Network Open just in the past week, looking at those who initially present with mild or severe COVID-19 symptoms, and how many have persistent symptoms 6 months after COVID-19 infection, which is shown in this yellow here. And you can see the fatigue, the loss of sense of taste or smell may persist in not an insignificant number of people for about 6 months. So while I am a huge proponent that the most important impact these vaccines can have is on severe disease and hospitalizations and death, we really can't minimize mild COVID-19 and, again, the longer term consequences of the infection. So what are the COVID-19 vaccines that are in late stage testing in the US through government sponsored development? And I show those here. We have the 2 mRNA vaccines, the 2 adenovirus vector vaccines, and then the 2 protein vaccines. The top 5 are all in phase 3 trials, and because you've heard data on the mRNA vaccines, these trials are going for at least 2 years. So those are interim data because we met a pre-specified number of endpoints, but again, those trials continue. The GSK Sanofi collaboration is the only one that is not yet in phase 3. That phase 2 is supposed to start today. So hoping that will happen. So let's look first at the mRNA vaccines. They're certainly the vaccines for which we have the most data. Probably everybody knows by now how mRNA vaccines work, but just to remind everybody, because there's some important messages for our friends and family and patients who ask some of these questions a lot. You know, basically we are taking the genetic sequence of a stabilized SARS-CoV-2 S protein. We are putting it in a lipid coat, in a lipid nanoparticle, and then injecting it, at which point it is taken up by both the muscle cells, but also preferentially tracks to the lymph nodes where it can be directly taken up by immune cells and antigen presenting cells. Then, of course, the mRNA never makes it into the nucleus. I like to make that point. People ask that a lot. Stays in the cytoplasm where the spike protein is made and processed. Then again, presented on the cell surface, looking like a viral infection and activating the downstream immune response. Many publications here. I suppose that's the other unprecedented aspect, is how quickly we are seeing these data make it into the public domain and make it into peer review. You know, I went back to the 2009 pandemic and there were immunogenicity studies, the initial immunogenicity studies being published a year or 2 years or 3 years after they were done. And certainly we have accelerated our ability to get important data in the peer-reviewed literature. We are, however, probably still dealing with too many data by press release situations, and even I am guilty in this presentation for using certain data that is only available through press releases or the New York Times. So everybody has probably seen this beautiful graph. I mix up the mRNA vaccines in terms of which I choose to use for my various examples. They are very similar vaccines and they seem to perform in very similar ways. So this is actually the Pfizer BNT162b2 vaccine, and you can see very nicely what we all hope to see anytime we do a vaccine study, which is we had steady accumulation of endpoints in the placebo group, which also tells us we had a lot of COVID-19 during the time we did these trials. And essentially after about the first 10 days, very few cases that accumulated in the vaccine group. And both the mRNA vaccines are about 95% effective against any symptomatic infection and 90 to 100% effective against severe disease. Now for this one, I'm using the VRPAC, the FDA briefing document, and I've chosen to use the Moderna vaccine just to illustrate that the fact that we did trials of 30,000 people or more and ensured that adequate numbers of subpopulations were included allows us to have some pretty robust vaccine efficacy estimates on subgroups, which I'm showing here. And for the most part, we are seeing high efficacy across subgroups. You can look at younger than 65, you can look at 65 and over, high risk, not high risk, race and ethnicity, and you see all very solid, very high estimates of efficacy against the primary outcome of any PCR positive symptomatic infection. And what about the reactions? Again, I'm using the Pfizer data to show the reactions, pretty similar with Moderna. We see a lot of pain at the injection site. Most is mild, some is moderate. And then in a not insignificant number of people, and for those of you who have received the vaccine, you're aware of this, you know, somewhere between 10 to 15% of adults may get fever or chills. A higher percentage, even the minority, have some fatigue in the 24 to 36 hours after receiving the vaccine, generally self-limited. Now, what about single dose efficacy? You know, this is a really hot topic. It's one of the most common questions I get asked here. And if you go back and remember this graph and look at this, you know, you can see that for the first about 10 days here, you don't have a separation. And that's probably for two reasons. One is because people may have been incubating the virus on day 0, and that's why they're getting recognized infection on days 1, 2, and 3, as you can see here. And the other is you simply haven't had time to mount an immune response. So we can start to look at the efficacy a little bit later, 10 to 14 days later, right about here, and see how these vaccines perform after a single dose. And if we do that for both the mRNA vaccine, and this is publicly available in the UK evaluation of their policy on these vaccines, and for the Pfizer vaccine in a letter to the New England Journal, we see that both of these vaccines, if you start counting about 14 days after the first dose, have about a 92% efficacy after one dose. So you see that here for the Pfizer, you see it down here for the Moderna. Now, we don't know how long that single dose efficacy will last because the vast majority of people got a second dose of vaccine at 21 to 28 days after the first dose. However, I would put forth that we don't have a 92% efficacy on day 28 and a 0% efficacy on day 29. There would be, if we only gave a single dose of vaccine, there would be some degree of efficacy that may wane over time, and we just don't know that degree of efficacy or how long it would last. And also, and data from both Mount Sinai and right here at the University of Maryland, we also know that people who are seropositive at baseline, and most of them were excluded from the primary outcome of these trials, do have a faster and more robust response after a single dose than people who are seronegative. And you can see that here in the orange. They also have, you can see it over here on the right side in the purple and the black colors. We also know that people who are seropositive do have more side effects after the first dose. So their first dose looks like the second dose for people who are seronegative. Fortunately, with new data from the CDC, they don't appear to have a lot of side effects after the second dose. So if you're seropositive, you seem to get more side effects of this vaccine after the first dose. If you're seronegative, you get it after the second dose. Just this morning, down the hall, We boosted our first participants in the Pfizer study with their third dose of vaccine. And Moderna has also started that just this week. So we will be able to see how people do both from an antibody perspective and from a reactogenicity perspective with a booster dose of these vaccines 6 months or more after receiving the second dose. So this is just a summary. I won't go over everything, showing what I've discussed here, that again, high efficacy against the primary outcome. And in terms of safety, I'll talk about this a little bit more. You know, we didn't see the anaphylaxis as part of the clinical trials. It's a rare outcome, and that's why we need to continue to assess these vaccines beyond the period of the clinical trials. However, the real challenge with these mRNA vaccines are their storage conditions, which again, everyone on this call is well aware of, having either to be stored at minus 70 degrees or in the freezer. So this has really limited, certainly globally, how these vaccines could be used. And this is just to make the point that assessment for safety and effectiveness does not stop at licensure. And in fact, it's through these robust safety surveillance systems that we have in the United States that we've learned more about the rare side effects of the mRNA vaccines. At present, our best estimate is about 2.5 to 5 million cases per million doses administered. And again, this will continue to be followed. So moving along to the adenovirus vector vaccines. So for these vaccines, again, they're still genetic-based vaccines. We're taking the genetic sequence of the virus. Instead of delivering it in a lipid nanoparticle, we are delivering it through an adenovirus vector, either a chimp adenovirus vector in the case of AstraZeneca, or an adenovirus 26 vector in the case of the Janssen vaccine. And I've already explained that the idea is similar in that we are, again, delivering this genetic code. It's also the S-glycoprotein. In the case of the Johnson & Johnson vaccine, very similar genetic sequence to what is used for the mRNAs, a 2-proline stabilized, pre-fusion stabilized S protein. The Oxford AstraZeneca vaccine is the only one that actually does not use that pre-fusion conformation. It's more the natural S-glycoprotein. So I won't say much about this. You know, again, safety and immunogenicity of these vaccines. They do induce neutralizing antibody, not quite as much neutralizing antibody as the mRNA vaccines. However, it's very difficult to control between labs, between assays, and we haven't really had good standards for this. We're relying on a comparison to convalescent sera, and it's really convenient samples of convalescent sera that these manufacturers use. But we are starting to get efficacy data on these vaccines. For the ChIMPAD, the AstraZeneca vaccine, we have had publications in Lancet, we have preprints, and just this morning, the Janssen or Johnson & Johnson, it's referred to in both ways, Ad26 vaccine, the briefing document from the FDA was released. I'll show a couple tables from that document that give us more information on the efficacy of that vaccine. So putting all 4 of these together on a table, I'll just emphasize a few points. One is that when you look at the efficacy against the primary endpoint, which is generally any symptomatic COVID, for Janssen it was moderate to severe, but in fact most of them were relatively mild COVID. We're not seeing the very high numbers that we saw with the mRNA vaccines. We're seeing about 67% for the Oxford AZ and about 75% in the US cohort for the Janssen. However, as I'll get to later, we're not really comparing apples and oranges here. The definitions were similar, but not the same. There may have been more variant strains circulating. We know there were more during the time of the Janssen vaccine. The real message here, and I will get into this, is shown right here. The very powerful effects of these adenovirus vaccines on severe illness and death. In fact, the Janssen vaccine is the only vaccine for which we have information on mortality. There were 5 deaths, and they all COVID deaths, and they all occurred in the placebo group. But really what could make these adenovirus vectors game changers, and particularly globally, is the fact that they can be stored for long periods of time at refrigerator temperature. And in the case of the Janssen vaccine, it's a single dose. So here's an example of some of the data from the AstraZeneca vaccine. This was presented publicly to the WHO. So this slide comes from that WHO presentation, where if they look at a WHO score greater than 2, which is pretty much any disease, you have about a 61% efficacy. But as you can see here, when we start looking at hospitalization, more severe hospitalization or death, and there was one death, so I misspoke on the Janssen. We had 5 in the Janssen, we had 1 in this trial. You can see that essentially, 100% efficacy against severe disease. And really exciting data that came out yesterday. This is only in a preprint, but the effectiveness of the first dose of COVID-19 vaccines against hospital admission during rollout of the vaccine in Scotland. So field conditions for using the vaccine. This was a prospective cohort study. It would be so wonderful if we had a system like this in the US. They have linked vaccination, primary care, real-time PCR testing, hospital and mortality records on 99% of the population in Scotland, which is about 5.4 million people. And they have rolled out there both the Pfizer vaccine, which you see here in blue, and the AstraZeneca adenovirus vaccine, which you see in pink. And the vaccine against hospitalization was 85% against the mRNA Pfizer vaccine, 94% for the chimp-ad vaccine, and importantly, 81% for both vaccines for people 80 years and older. There weren't a lot of people 80 years and older for these 2 vaccines in the trials. So again, this is only a preprint. I look forward to seeing the peer-reviewed data, but certainly very promising results. We also have promising results out of Israel. I have those in my backup slides, and if we have time or people have questions, we can also go through the impact that we're also seeing with the Israeli rollout of these vaccines. Now, until this morning for the Janssen vaccines, I didn't have public data that I could show you. I only had this news release where they show, again, the single dose being 66% efficacious at preventing moderate and severe COVID, but much higher in preventing severe disease. But with the FDA release of the information, we can also get a look at the efficacy by subgroups for the Ad26 Janssen vector vaccine. I chose to use the table in that FDA document that looks at severe or critical disease. And you can see here pretty robust numbers across the subgroups, anywhere, wide confidence intervals from 75 to 100% effective. The one subgroup that might be of concern, and I would expect some discussion of this group at the FDA meeting on Friday, is this over 60 years of age with comorbidities. Again, wide confidence intervals, but does have a point estimate that's somewhat lower than the others. This trial is still going in the same way that the other trials are still going. These trials are continuing to accumulate endpoints, although for the mRNA vaccines, the placebo recipients have already been vaccinated. So we do have a finite period of time, though, where we'll continue to accumulate endpoints and perhaps get more robust estimates for some of these smaller subgroups. And then finally, I just want to say this is also in the FDA document about the Ad26 Janssen vaccine, is that this occurred at a time in South Africa where the concerning 1351 variant was circulating. I'm going to talk in a minute about the variants. In fact, about 67% of their cases were sequenced, and of the sequenced cases, 95% were the variant strain. So we are seeing that efficacy in South Africa of this vaccine in the face of the variant strain. So that is definitely good news. So what about the protein vaccines? You know, protein vaccines are our tried and true formulation for vaccines. We know they work very well, but we know that they also take longer to manufacture. It's harder to make a protein with the purity that the FDA requires. So Novavax really has terrific neutralizing antibody data in their phase 1. They have a nanoparticle with a matrix M adjuvant, very well tolerated, again, high neutralizing antibody. The phase 3 in the US, which is led nationally by Karen Kotloff here and locally by Monica MacArthur, just did a terrific job in recruiting in a very short period of time and in face of other EUA vaccines being rolled out. So we hope that we will have data on the Novavax vaccine in the US in the coming weeks to months. In the meantime, Novavax did 2 smaller trials in the UK and South Africa. You know, they showed in the UK an efficacy of about 90% with a variant efficacy of about 85%. In South Africa, the overall efficacy was about 50% in the face of that variant. It went up to 60% if you excluded HIV infected. So again, these variants are certainly something to keep an eye on. And so I'd like to talk about the variants. This looks at the variant that was first recognized in the United Kingdom, but as you can see from this map from early February, has now spread throughout the world. So this B117 variant, you can see, has been recognized throughout most of the US. It is highly likely it's in these states in white, but we have not had a very high rate of sequencing in the US until very recently. You may have seen Governor Hogan's announcement this morning about a partnership with us here at the University of Maryland, as well as at Hopkins, to greatly increase the number of viruses we are able to sequence in Maryland. And much more concerning is actually the 501YV2 variant that was first recognized in South Africa, that also has been recognized in the US, although more sporadically, but we had one of the first cases here in Maryland, and you can see, again, the more sporadic nature of that virus. So what do we know about the immune response to these variant strains? Well, we know that there certainly is diminished neutralizing activity of the mRNA vaccines against that B1351 variant that was first recognized in South Africa. These were publications, both correspondences in the New England Journal from about a week ago, looking at the mRNA vaccines, the Moderna and the Pfizer here, and you can see the neutralizing antibody is diminished. This is the baseline vaccine strain. This is the 1351 variant, similarly here. Much more preserved for both vaccines in terms of the variant that was first recognized in the UK, that B117 variant. However, even with these diminished neutralizing responses, we're still seeing neutralizing titers above baseline, far above baseline. And so again, without a correlate of protection, it's difficult to know how these vaccines will perform against these variants. And that's obviously a very, very important topic on the research agenda going forward. And perhaps the last big question that I get asked a lot is, do vaccines affect transmission? And I might turn that around and say, do vaccines affect viral shedding? Getting from viral shedding to transmission is not necessarily linear, and it's certainly much easier to measure if there's virus in the nose than to say, is that virus in the nose also being transmitted and infecting somebody else? So just a couple slides on this. I thought this was a very interesting study looking at any SARS-CoV-2 PCR positivity in relation to, can you culture the virus? And you can see early in the illness, the positive cultures are here in this red dots, and then the blue are the negative cultures. The open dots, we don't have a culture test. But you can see, as expected, you have more virus that's being able to be cultured early in an illness. It goes down over time, and then you have a lot of PCR positivity later in the illness that may actually not represent viable virus. So an important concept to keep in mind as we translate a PCR virus in a swab to the potential for transmission. So in terms of protection against asymptomatic infection, we don't have a lot of data. The data that we have are in the right direction, meaning we are seeing fewer positive swabs and less virus in vaccinated people than unvaccinated people. One example is this table from the FDA submission by Moderna on the mRNA 1273, where they simply swabbed the noses of everyone when they came in for their second vaccine dose, and showed that if you were negative at baseline and then had a positive, the vaccine reduced a positive swab at your second dose by about 64%. Again, we don't know if any of this virus is viable virus, if it's being transmitted, but if it's not in your nose, it is unlikely to be transmitted, assuming highly sensitive tests, which we believe these PCRs to be. The other data that we have looked at quantitative PCR. So the higher your CT value or cycle threshold, in fact, the less virus you have. And the lower your cycle threshold, you have more virus. And so we're picking it up with fewer cycles, we're picking it up sooner. And this is completely an ecologic study in Israel after rollout of the vaccine, but really quite interesting that as the vaccine is being rolled out in the older adult population, which is seen in blue, it appears that the viral quantities are going down in that same population. So again, there are a few studies now, I haven't shown all of them, that suggest these trends toward less viral shedding. That being said, there is still virus, as I've showed you. We need to tell people to keep wearing masks. They could still spread this virus, but obviously a very important area of research to understand how vaccines can impact viral shedding. So just a quick table here to perhaps summarize a lot of what I've said, and I worked on this with Dr. Rapaka and Dr. Hammershade, two early career scientists in my group, in terms of, when we look at comparing and interpreting these estimates of efficacy in trials of COVID-19 vaccines, we can't just compare a single number. We need to understand the context. What was the study population? Did it include older ages? Did it include HIV infected? I showed you an example where that affected an efficacy estimate. What about the virus characteristics? Was a variant strain circulating during the time of the trial? What was the force of infection? Were attack rates high? Were people being exposed many times to a lot of virus? How did we define our endpoint? When did we start counting our endpoint? Did we start counting at day 0, or did we start counting at day 10 when people get an immune response? And then, was there differential use potentially of treatment and preventives? We think this is probably negligible at present, but could be more important for future trials as we see more use of monoclonal antibodies and perhaps antivirals. So just in the last few minutes here before questions, I'll give you a little bit of an update on how we're doing with COVID vaccinations in the US. Here's a picture of the US. The darker your state, the more people per population you have vaccinated. So you can see, West Virginia doing very well, Alaska doing very well here. We've had, as of 2 days ago, when I pulled this map, 44 million people who have received 1 dose of vaccine, and pretty evenly split between the Pfizer and Moderna, about 20 million receiving the 2nd dose of vaccine. Unfortunately, we are seeing what we think is either some access bias or perhaps hesitancy in that there are more white people receiving vaccine than they are represented in the general population. We see here, Blacks about 6.4% of all vaccines administered, Hispanic about, here it is, 8.7%, so lower than their prevalence in the population. So we know we have some work to do in terms of equity, because of course, these are the populations who get more disease, get more severe disease, and are working in many of the occupations that are vital and can't be accomplished through telework. So we have some work to do on the equity front in the US. And some of this is not surprising. This was a survey that was done. This is on the NFID website, just saying that among Black adults, they're much less likely to say they will get the vaccine. We know they have more vaccine hesitancy, more misconceptions. So again, it's going to take some work to overcome these barriers and this vaccine hesitancy. What about around the world? Again, the bluer the country, the more vaccines that have been introduced. And you can see here in this darkest color is Israel, which is leading the world in terms of vaccination per number of vaccines administered per 100 people. Here we are in the US, not doing too badly, but Israel, United Arab Emirates, UK are above us. And as expected, really no lower and middle income countries on this chart. Just this morning, vaccine arrived in Ghana through the WHO COVAX efforts. So that is a major achievement. And not even reflected yet here is during this past week in South Africa, Janssen actually donated vaccine to begin vaccinating healthcare workers in South Africa. So you can see that's somewhat represented here, and pretty soon we will have a blue color here on Ghana to reflect the beginning of their vaccination campaigns. So in summary, we need safe and effective vaccines that are accessible, affordable, and available. We are in a critical supply shortage right now, certainly in the US and most definitely in the world. We have quite robust safety and efficacy data from these early clinical trials. We have post-introduction impact in Scotland, in Israel being demonstrated, and we have an early indication of impact on viral shedding, but that's a to be determined in my mind. We have safety systems and processes in place, and these variant strains are really our latest challenge. I do want to emphasize that these variants emerged in the absence of vaccine. So we should not be making the correlation between their escaping vaccine immunity. They emerged in a setting of unmitigated viral replication and transmission. So this is an even further impetus to roll vaccines out as quickly as we can. You know, there are neutralizing antibody responses. They're not quite as good as to the vaccine strain, and we've seen efficacy in certain settings against these variant strains. But as risk mitigation, we're already looking at new strains, variant strains that will be in testing very soon. Mix and match, heterologous prime boost. Are there other ways that we can get this? You know, maybe non-S-based vaccines that would have more heterologous protection. And if we didn't do it before because it's the right thing to do, again, these variants are showing us that if we just protect the US population, we are never going to be out of this mess of a pandemic. And we need a true global surveillance and vaccination plan. And so it's really, to me, wonderful progress that the US currently appears to be, again, fully participating in global activities in World Health Organization efforts to get vaccine to all. So thank you for your attention, and I'm happy to take questions as time allows. How do you want to handle the questions? Do you just want me to read through them?
Hi, Dr. Neuzil. I'm actually going to go ahead and read through all of them since we have several throughout your talk. So the first one I have is from Dr. Hochberg. Can you comment on the similarities and differences between long COVID and chronic fatigue syndrome?
Yeah, to be honest, I'm probably not the expert that can comment on that, and it would be better for others to do so. However, you see what I think are some of the best data we have so far, the prospective data that I showed you over that 6-month period of time. Numbers are still fairly small. Again, as I emphasized, it appears to be predominantly fatigue, loss of taste and smell, and nonspecific symptoms such as headache. So I think time will give us a better indication of where that's heading.
Got it. The second question is from Anuj Gupta. How do you do a phase 3 trial if you already have effective vaccines? Do you have to do it compared to approved vaccines?
Yeah, so that's a great question. So early on, we did not have that problem. So with the first trials, actually, essentially all of the trials except for the Novavax trial got started before we had the availability of vaccine. Once you have availability of vaccine, it does become very difficult. It becomes an ethical dilemma. For the mRNA vaccines, what we did is we offered vaccine to all of the placebo groups. For the adenovirus vector vaccines, similarly, if a participant wanted to be unblinded and receive an emergency use authorization vaccine, they were allowed to do so. Of course, because we were still in a critical vaccine supply shortage, you know, there were a fair number of people doing that, but for example, the people in the trial that weren't in a category that was able to receive vaccine yet stayed in the trial. For the Novavax vaccine, it was just an issue that was discussed during the informed consent process. It was a 2-to-1 randomization. And again, the mix of participants in that trial is slightly different. There are about 13% of people who are over age 65, which is down a bit from where the mRNAs were. And if you had other high risk conditions, you would probably choose to get an EUA vaccine. That will likely be the ongoing Novavax trial, the last one that is able to be done as a fully controlled, placebo-controlled efficacy trial. So future vaccines will likely either be comparative trials or they may be done in countries that have different recommendations and use vaccines in different ways. So I think in the same way we look at influenza vaccines, that's probably what you will see going forward.
Thank you. The next question is, how would you respond to people who don't see the point of getting the vaccine because it doesn't guarantee that a recipient won't get COVID?
Yeah, so my main response to that is that it will keep you from getting severe disease. It'll keep you from being in the hospital with disease. It'll keep you from dying. And you know, while not getting a cough and a cold and losing your sense of taste and smell is certainly a nuisance or beyond a nuisance. It's not what is filling up our hospitals. It is not what is keeping us in lockdown. If this virus caused a common cold, then we wouldn't call this a pandemic. We'd call it a rhinovirus. So it's because this virus causes severe disease. That is a requirement to call a pathogen a pandemic pathogen. So I reassure people that these are highly, highly effective vaccines, that even for the mRNA vaccines, you're not going to get much better than 95%. And then my second response is that the more immunity we have, you know, this is not tetanus that's in the soil and I am exposed as an individual. The more people that get immunity from vaccines, the more it benefits all of us and the faster we can get back to normal life.
Got it. The second question is now, could you comment on COVID-19 in pregnancy and especially vaccination in pregnant women, concerns for embryo, fetus, SOPs, gaps of knowledge?
Yeah, so that's a great question. That's something that I and others have been lobbying for: more data, more inclusion in a well-informed way of pregnant women in these studies. I actually saw the summary the other day, but I have to tell you, I'm going by memory. There were maybe about 60 pregnant women total included in these mRNA studies. There were no safety effects out of the ordinary. All of these companies have done development and reproductive toxicity or DART studies in animals and seeing no problems in animals. That being said, the CDC has a very robust safety surveillance system for pregnant women, and we will soon be sponsoring an NIH trial that also looks at pregnant women and the effect of maternal antibody transfer to the infant and infant outcomes. You know, you really don't have a vaccine for healthcare workers and first line workers if you don't have a vaccine for pregnant women. So both the ACIP and the American College of Obstetrics and Gynecology, based on the data that we have so far, which isn't a lot, have given a permissive recommendation for pregnant women to receive this vaccine.
Thank you. The next question is there any data yet on immortalized memory B and T cell and plasma cells pointing towards long-term immunity?
Yeah, so that's a great question. We are getting more of those data now. In fact, some of the inconsistencies with the amount of neutralizing antibody, for example, we're seeing with the Janssen Ad26 vaccine, which isn't particularly high, but their high efficacy even against the variant strains in South Africa suggests that there is a robust T cell response. And in fact, we are seeing robust T cell responses in animals. So this is being actively studied, and we will learn more about this over time. You know, one corollary of the mRNA vaccines being so highly effective is that to look at correlates of protection, and whether that's T cell or B cells or antibody, you have to have some vaccine failures. And you have to statistically have about 30 vaccine failures to be able to even start those analyses. And, you know, in the first trials we had, again, they were so effective, we didn't have enough vaccine failures. But there is a very robust program across the trials to look at immunogenicity and correlates of protection.
Thank you. The next question is there any information on the immunogenicity caused by the different doses between Pfizer and Moderna? Do we know if the higher dose will offer protection, longer protection, or cause more side effects?
Yeah, so that's a really good question. You know, in both of those vaccines, we had dose ranging that was done in phase 1. That the dose ranging picked the highest dose that was well tolerated, that gave you a good antibody response. Because it was a pandemic, we didn't really take the time to optimize. You know, you may have in a different scenario looked at even more different doses, and you may have chosen differently. What do I mean by that? Moderna, subsequently to entering the big phase 3 efficacy trials with 100 micrograms, did do a second study comparing 50 and 100 micrograms and found very similar immune responses. So going forward, they are working with the FDA. We don't know the outcome of that. You know, you may eventually see a 50 microgram dose for the mRNA vaccine. So again, we have duration of immunity out to about 6 months with these mRNA vaccines. It looks quite good, again, in a test tube, in an assay, out to about 6 months. The 50 microgram data is a little bit behind that. So we're learning as we go, but it's possible you would see dose adjustments going forward.
Got it. We actually have several more questions. Do you have time to answer them? I understand it's 1.
Yeah, I honestly have to look and can't remember.
No worries.
I have at 1. I do have, it's actually our a meeting for our maternal study that I was mentioning. So I'll stay on a couple more minutes, but I probably shouldn't stay on much longer than that.
Sounds good. We'll pick up with 2 final questions then. The second to last question is, fantastic presentation. Thank you, Dr. Neuzil. Was wondering, in light of older adults being affected the most with an 80 percentile effectiveness, the data seem to always present a 95% for everyone else, while the patient who is essentially and really affected is the older adult, especially 80 plus, with an increasing aging population. I understand the concept of the herd immunity, and we are hoping to get there. Why do you think this is? Thank you again.
Yeah, well, I think, you know, we know that frail older adults generally don't have as much of a robust immune response. You know, for the most part, we are really seeing quite good efficacy. Again, if you compare it to influenza or certain other vaccines in older adults, and it does seem to attenuate illness in older adults. This will be my plug for older adults, in that we are underusing the monoclonal antibodies. If we are seeing older adults with early infection, mild infection, whether in a nursing home or as an outpatient, we should be using these monoclonal antibodies, whether they have been vaccinated or not, as an adjunct in preventing severe disease.
Excellent. And the final question is, what is the scientific rationale for waiting at least 90 days to receive vaccine after a monoclonal antibody or convalescent plasma treatment? And do we know whether natural immunity is affected / not as long lasting after either of these treatments?
Yeah, so that's a great question. That's another high research priority. You know, that's based on the theoretical concern that if you have passive antibody, it will interfere with your ability to generate active antibody from the vaccine. So the thought is that if you look at the half-life of these monoclonals, you will be protected for a period of time. When the antibody starts to wane and you might start to be at risk for disease, would be a good time to vaccinate. Again, this is all theoretical, and those studies are being done now, but we don't have actual data to show what the interference might be and the absolute best time to vaccinate people who have received passive antibodies.